You know, we've always looked at fossil bones as these amazing windows into the deep past, right? They tell us stories about incredible creatures that roamed the Earth millions of years ago. But what if we could get way, way more detail out of those stories? What if we could learn things we never thought were possible? Okay, let's dive into this.
So, get this. Here's a question for you. Could we give a 3 million-year-old antelope a blood test? I know, it sounds completely nuts, like something straight out of a sci-fi movie. I mean, how on earth could you analyze the blood of an animal that's been nothing but fossilized bone for millions of years?
Well, believe it or not, that totally impossible-sounding idea is actually happening right now. Thanks to a revolutionary new field of science, what was once pure fiction is now a reality, and it's completely changing how we see ancient life.
And it all has this really cool name, paleometabolomics. Let's just break that down real quick. Paleo, means ancient, and metabolomics is the study of metabolites. These are just the tiny chemical byproducts left over from an organism's metabolism, all the processes that kept it alive. So basically, we're studying the chemical ghosts of life that have been preserved in fossils for ages.
Okay, so how in the world does this actually work? You have to wonder, how can these super delicate chemical signals possibly survive for millions of years, locked away inside solid rock? Well, the secret, it turns out, is in the very structure of bone itself.
See, as a bone is forming in a living animal, it's not just a solid, impenetrable thing. It actually traps tiny little molecules that are floating around in the bloodstream at that moment. So it basically becomes this perfect molecular time capsule, capturing a chemical snapshot of the animal's life.
And here's the really key part, the reason this all works. The little spaces where these clues get trapped are nanometer-sized. I mean, they are so unbelievably small that even bacteria, which are way bigger, can't get inside to eat them or break them down. And that is how these chemical clues can stay perfectly preserved for millions and millions of years. It's wild.
So how do you get them out? Well, the lab process is actually pretty neat. First, scientists take a tiny piece of the fossil and dissolve it in some weak acid. Then they pop that sample into a centrifuge and spin it really, really fast. That pushes all the lightest molecules the metabolites were looking for right to the top. This liquid soup at the top is then carefully injected into a machine called a mass spectrometer, which is like a super precise scale for molecules. It weighs everything in the soup, and finally, they compare those weights to a huge library of known compounds to figure out exactly what they've found.
All right, so the scientists have found all these ancient molecules. Now the real detective work begins. What stories can these tiny little chemicals actually tell us? Well, turns out the clues fall into two very different but equally cool categories. On one hand, you've got what are called endogenous metabolites. Think of these as clues from inside the body. They can tell you about the animal's health, if it had any diseases, stuff like that. And on the other hand, you have exogenous metabolites. These are clues from outside the body, mostly from the plants the animal was eating. And these clues tell us all about its diet and the environment it was living in.
So to put this all to the test, researchers used this technique on some seriously old fossils. We're talking a 3-million-year-old antelope from South Africa, a 2.4-million-year-old elephant tooth from Malawi, and even some rodents from Tanzania that are almost 2 million years old. And what's really cool is that these are all sites where our own early human ancestors have also been found.
And the results? They were absolutely stunning. For instance, they found an internal clue, a metabolite from the parasite that causes sleeping sickness today. Think about that for a second. They were able to diagnose a specific disease in an animal that died millions of years ago. That's incredible.
But this is where it gets even more fascinating. That one little clue about a disease, it also paints a huge picture of the entire environment. Why? Well, because that parasite is carried by the tsetse fly. And the tsetse fly is super picky. It can only survive in very specific conditions. So finding that parasite tells you that the right kind of habitat had to be there too. The animal's health is a direct window into its ecosystem.
And the external clues, the ones from the plants, they back this all up. They showed that these ancient African landscapes were way different than they are today. We're talking significantly wetter, warmer, and with a lot more forest shade. It points to this mixed tropical habitat that had really distinct wet and dry seasons.
But while they were doing all this amazing research, the scientists stumbled onto something else, something they weren't expecting at all, a discovery that would completely flip a long-held scientific assumption right on its head.
For decades, whenever paleontologists found a fossil,
The soil packed around it, what they call paleosol, was always just considered, well, a contaminant. The idea was that any chemicals from the soil that might have seeped into the bone were just noise, something you had to clean off and ignore to get to the real data.
But when they actually analyzed the ancient soil and compared it to the metabolites inside the fossil, they found a massive overlap. And this led to a jaw-dropping realization, which this quote sums up perfectly. The ancient soil, the paleosoil, reflects the lives of all the organisms living there.
So here's the big twist. The soil isn't contamination at all. It's completely connected to the animals. As plants and animals live and die on a piece of land, their metabolites become part of the soil. The soil isn't just dirt, it's a shared chemical library of the entire ecosystem. It's like a logbook, chronicling everything that lived and grew there.
So what does all this really mean? I mean, this discovery is huge. It's not just some new gadget for the paleontologist toolkit. It gives us a way to reconstruct these ancient worlds with a level of detail that we've never, ever had before.
This new kind of molecular ecology can tell us some incredibly specific things about a prehistoric habitat. We're talking about the soil's pH level, the minimum and maximum rainfall, the minimum and maximum temperature, even the exact type of tree cover, and the elevation above sea level. It's just an insane level of detail.
The potential here is absolutely mind-boggling, right? It suggests that one day we might actually be able to describe the world our earliest ancestors lived in with the exact same precision we use to describe a modern rainforest or savanna today.
It basically transforms a fossil from being just the remains of a single animal into a rich chemical library of an entire lost world. And it leaves us with this really thrilling question. If we can learn all this from just a few samples, what other ancient stories are out there just waiting to be read?